my curious little champions, I welcome you all to score 99% in 99 days. Today we are going to solve certain very important previous year questions from the chapter that is how do organisms reproduce, and with that we are going to cement on all the concepts in our brain correctly. So I always say without practice you are not actually studying. So you studying concepts and all is a very different thing. Now you applying it to while solving questions is a whole another world. So you have to know how to tackle both of them, and for that we are going to help you. So let's get started over here. The first question that we have for the day is, what is this? The number of chromosomes in parents and the offsprings of a particular species undergoing sexual reproduction remains constant due to. So they are saying the parents, suppose this is a cell, and in that the number of chromosomes is 46. Now in the offspring which is being produced, in their cell also the number of chromosome is 46. How can that be maintained? So you need to have two parents. This is parent number 1 and there will be parent number 2. If parent number 2 also have 46, then 46 plus 46 the child should be having 92. But how this comes to be 46 exactly? So what happens during gamete formation, children? This parent has to form a gamete. This parent also has to form a gamete. What about halving the number of chromosomes during gamete formation? What about halving the number of chromosomes during the gamete formation? So if we say that I have a pair of chromosome, this is this is the first pair of chromosome, then we have second pair of chromosome, and together each pair, if we see together, we say we have 46 number of chromosome. But if we talk about pairs, how many pair do you have, children? 23 pairs we have. In that we have 1 and 2. Similarly, how many chromosomes we have? 46 chromosomes. In each pair there are 2 chromosomes, right? So in total, if we say individually, if we count 1, 2, then 3, 4, then 5, 6, we have 46 of them. What about we halve the number of chromosomes? We only send half of the number of chromosomes in a gamete. If we do that, then the gamete cell will be having 23 chromosomes. Agreed? From a pair, if I am sending each. Okay, so I will be having 23 chromosome over here. This gamete will be also having 23 chromosome. Now, when this gamete and this gamete fuses, what will I get? I'll get 46. So this is the exact thing that happens. That is, during the gamete formation, we halve the number of chromosomes with the help of a process or a type of cell division which is known as meiosis. So in sexual reproduction, during gamete formation, which type of cell division takes place? It is meiosis, my dear children. Okay, so number of chromosomes in parents and the offsprings of a particular species undergoing sexual reproduction remains constant due to what? Doubling of chromosome after zygote formation? No. Halving of chromosome after zygote formation? After zygote formation, why do we need to do so? No. Doubling of chromosome before gamete formation? No. Halving of the chromosomes at the time of gamete formation. While we form the gametes, we have to halve it. So this becomes your answer. Cool? Now the next question. During adolescence, reproductive phase starts, and what happens? During adolescence, the reproductive phase starts, and then what happens? General growth rate begins to slow down, height becomes less, the body weight is reduced, hair growth decreases. No, I would rather say general growth rate begins to slow down because you are in a reproducible age. So all the other things, your reproductive organs and everything start to grow. So more concentration is put to that particular angle, okay, rather than the general growth rate. So this kind of begins to slow down. I'm not saying completely goes off. No, it kind of slows down, my dear children. Okay. Next, which one of the given statement is incorrect? Among them, which of them is incorrect? DNA has the complete information for a particular character. Yes, our DNA has all the information because our DNA has certain segments which you call as what, children? You call them as genes. And now these genes are responsible for coding what? Protein. These genes are responsible for coding protein. So DNA has complete information for a particular character. It's true. DNA is molecule responsible for inheritance of characters from the parents to the offsprings. It is also true. We inherit DNA from them to us, and that's how we inherit the character. What are the character? Basically, in them and in us, we have a common protein. Okay, they are going to give us these genes. These genes are going to make the same proteins in our body. These proteins are going to give us the same character. So that is why we say the characters we get it from our parents. This is actually the genetic material that we get from the parents. Now, change in information will produce a different protein. Obviously, if the genes are altered in some or the other way, it would make some protein, some other protein, probably protein number 2, which would have a different effect, right? Now, that is also correct. Characteristics will remain the same even if protein changes. Do you think if protein changes, the character will remain the same? No. Probably this protein might be coding for high amount of melanin, which is why my skin color will be dark. If there was a protein which was coding for less amount of melanin, my skin shade would be less. So the character is being changed. So characteristics will remain the same even after protein changes. That is a wrong statement. And you were asked for finding what? Incorrect statement. Always, whenever this type of question comes, no, underline what you have to find. Because sometimes we try to find the correct statement when you come out and oh, all of them are correct. So underline, this becomes a lot more easy for you to solve the question. Okay, now next. Choose the correct order of the stages of binary fission in leishmania. So leishmania, okay, basically it is something that causes kala azar, sleeping sickness as well. Okay, so how do they divide? You see over here they have flagella, they make one more one. Okay, they make one more kind of it. The nucleus also divides. After the nuclear division completes off, there will be a slow slit coming towards upper angle. So there is a longitudinal axis through which they kind of bifurcate their body into two. So this is the absolute way of division: 1, 2, 3, 4 and 5. This is in the correct order. So we have to say 1, 2, 3, 4 and 5. This becomes the answer. Okay, at last they are going to divide into 2. Cool? Chalo, next. Consider the following three flower, namely X, Y and Z. Which flowers would develop into a fruit? Which among them do you think will develop into a fruit? Will X develop into a fruit, my dear children? For a fruit, what do you need? What develops into a fruit? What develops into a fruit? It's the ovary that develops into a fruit. So if you know the structure of the female reproductive system of a plant, it looks like this. So this is the stigma, this is the style, and this is the ovary. Inside the ovary, what do you have? You have ovule, and inside the ovule you have the female gamete. So the ovary is the one that will be forming the fruit, and ovule is the one that will be becoming a seed when the male and the female gamete fuses. When the male and the female gamete fuses, what will you have over here? Instead of the female gamete, you will be having the zygote over here. And it has to be protected, it gets protected, okay, with the help of a seed covering and everything. Well, this particular portion which has the zygote within it, what do you call it? You call this now as a seed. And outside that, the ovary will be turning into a flower. So over here, you see the ovary here, you see the ovary over here. Do you see any ovary over here? No, it just has the male reproductive system. So which flowers would be developing into a fruit, my dear children? This and this can. So X and Y can only develop into a fruit. None of the others would be developing into the fruit. Cool? Next, the thread-like structures that develop on a moist slice of bread in rhizopus are. So basically, the thread-like structures which are going to form over here, okay, it will be many in number. They will come together to form the mycelium sort of thing. What do you call this thread sort of structure? You call this as hyphae. When the hyphae comes together, it is going to be known as mycelium. This is going to be known as sporangium. Over here, what do you have? You have spores, my dear children, stored inside this. Okay, these are root-like structures which are being formed. This is also going to be made up of hyphae only. Everything is going to be made up of hyphae. So this becomes your answer. Okay, can you see this with the naked eyes? No, you need a microscope for seeing it. Next, assertion says, testes in human males are located outside the abdominal cavity in the scrotum. So suppose this is the abdominal cavity and the testis which is there, it is located outside like this. It is located outside like this. Why is the testis located outside? Because first thing, inside, if this is the abdominal cavity, the temperature is way too much. And testis is a place where what formation takes place, children? Sperm formation takes place. Now the enzymes which are needed for sperm formation and everything will be only active at a temperature 2 to 2.5 degrees Celsius lower than the normal body temperature. You get it? Which is why the testis is not freely located like that. It is inside a sac-like structure which you will be calling as what, children? Scrotum. Okay, so your answer becomes testes in human males are located outside the abdominal cavity in the scrotum. This is correct. Scrotum provides a lower temperature than the normal body temperature for sperm formation. This is also correct. So can I say assertion and reason is true and the reason is explaining the assertion as well? Yes. So assertion and reason are true and the reason is the correct explanation of the assertion as well. Okay? Chalo, next. Now we have entered the 2 marker corners. So this time we have to write the answers and understand things very well. So give reasons. Placenta is extremely essential for fetal formation. Do you think placenta is important for fetal formation? How do you think placenta is being formed? So basically the zygote, which is single celled, it divides. Okay, now you have 2 cells, then it will be having 4 cells and so on. So there will be multiple cells over here and what do you call this structure as? Embryo. This embryo will slowly go and get embedded in the wall of the uterus. So what this wall known as endometrium, the innermost wall which is red in color because of the blood vessels and everything. Okay, now slowly it will get inside, it will get implanted over there. So from the embryo side, okay, from the I was going to fall down. From the embryo side, they are going to make certain structures like this, finger-like structures like this. What do you call this as? And here the embryo will be forming somewhere. Okay, and it will also form certain finger-like structures. You call them villi. You call it villi. And now this villi is supplied with certain blood-filled spaces. There will be certain blood-filled spaces. So where is this blood coming from? Blood is coming from the mother's side. Blood is coming from the mother's side. So this thing that you see over here, the villi and the blood-filled spaces together, you call this as placenta. So the embryo is also helping in the formation of placenta. The mother is also helping in the formation of placenta. Okay, the blood vessel which comes from the mother gives blood in the blood-filled areas. From here, with the help of diffusion, things gets inside the baby's body. Okay, through the umbilical cord and stuff like that. I'll be showing you. So why do you think it is important? Why placenta is important? Because there is a transfer of things going on. Okay, basically the mother is able to provide what? Oxygen and every important gas to the baby. The baby, which is giving out carbon dioxide, it has to be removed, otherwise it becomes toxic. So that CO2 is being exchanged to the mother and from the mother's body it is being given out. The waste materials which is given in the baby's body, that is also expelled out. Okay, so that is the use of what? Placenta. It is the use of placenta and also it provides nourishment and everything. You get it? Now that is the thing. I will be showing you a picture and I will be also showing you how you have to write the answer. Now there is one more question. Uterine lining becomes thick and spongy after fertilization. So after fertilization, the uterine lining, for example, this is your uterus. And its lining, it has 3 different layers. Outer is perimetrium, then for outer one is perimetrium, okay. Then is myometrium, which is muscular layer that will help in contraction. So during baby's expulsion of moving out, basically the procedure moving out, okay. At that point the muscles have to contract, that's when the baby will move out. And then there is a layer, and then there is a layer, okay, which is glandular. And the question is asked about this layer only. Uterine lining becomes thick and spongy after fertilization. So this uterine lining becomes very thick and spongy after fertilization. What do you mean by after fertilization? When the ovum and the sperm, they fuse, after it, this particular thing that you see, it becomes thick and spongy. Why? Because now children, you are going to form what? Zygote. The zygote is going to form into an embryo. This embryo is now going to come and basically embed over here, get implanted over here, and gets its nourishment from this particular layer during the initial few time, and then the placenta is being formed. So the initial source of nourishment is what? The endometrium, this particular layer. What is it? Endometrium. So how do you write this answer? First thing, this is the placenta that I was making. So there is baby over here, okay, just the embryo, oh, okay. There is the embryo formation still taking place somewhere over here, okay. It will have the umbilical cord and everything, okay. You have seen the navel of the baby, right? You have seen the navel of the baby. So probably I should be making like this. Wait. So this navel is connected like this. The umbilical cord is connected. Umbilical cord has lots of blood vessels, okay, which goes into each of the chorionic villi, the finger-like projections. Now the chorionic villi is surrounded by what? Blood spaces. In this, who is providing blood, children? The blood vessels which are coming from the mother. From here, the blood is going in there. From here, the blood is going in there. From here, whatever O2 or whatever thing is there, it has been transported into the blood vessels, and this blood vessel is going to go back into the baby. And there the things happen. So diffusion, direct contact of blood is not happening over here. There's diffusion that is happening over here. Oxygen is being diffused from here to the blood vessels, and that is being taken back to the baby. Okay. So the developing embryo gets nutrition from the mother's blood with the help of placenta. Okay, you were asked why it is extremely essential, because it is going to get nutrition from the mother's blood with the help of placenta. Placenta contains villi on the embryo side of the tissue and blood spaces on the mother's side surrounding the villi. Yes, the embryo is providing villi and the mother's side is providing blood spaces. These villi provide a large surface area that facilitates passage of nutrients and oxygen to the embryo from the mother through the blood. Waste substances produced by the embryo are removed through placenta into the mother's blood as well. Whatever waste ions are there, they will move out. The same thing also. Okay. Now, uterine lining becomes thick and spongy after fertilization. Every month, one egg is released by the ovary and uterus prepare itself to receive a fertilized egg. As a result, the lining of the uterus becomes thick and spongy. This is required for nourishing the embryo after fertilization. So the question is kind of wrong. Spongy after fertilization, it's before fertilization only. Before fertilization only, it will start to become thick. Okay, so that if fertilization happened, then this guy has to be providing nourishment. If fertilization is not happening, then what happens, children? This layer sheds down, and this is known as menstruation. This is known as menstruation, or basically what you call as periods. Remember, the whole layer is not shedding down. A basal layer still remains. The other layer, it falls down. There's a temporary layer being formed that falls down. Now, you don't say falls down, you say sheds down. Okay, so this is how you have to write the answer. Please write in points, otherwise you're going to lose your marks. So probably from here to here becomes your first point, then here to here becomes your second point. Cool. Next question. Name the reproductive and non-reproductive parts of bread mold rhizopus. List any two advantages of vegetative propagation. I'll start with any two advantages of vegetative propagation. What do you mean by vegetative propagation? Where is the asexual mode of reproduction in the case of plants, where the plants are going to reproduce with the help of vegetative parts. Okay, with the help of vegetative parts. For example, usually the plant produces with the help of the flower parts, right? But now it is going to reproduce with the help of stem, or probably root, or probably leaf. Okay, for example, use of bryophyllum. The bryophyllum has kind of buds being formed over here. And these buds, when they fall off, they are going to make a new plant. So here the leaf is a reproductive organ. But here, do you see fusion of gametes and everything? No fusion of gametes is being seen over here. So that is vegetative propagation, where the plants are reproducing using their vegetative parts, and there is no fusion of gametes. Nothing is happening like that. So what are the advantages? First thing, even seedless type of plants can reproduce. Okay, even seedless, because over here we do not need seed. When a seed needed, when fertilization everything is done, that's when you have seed. Okay, and this seed gives rise to a whole new plant. But over here, in the case of vegetative propagation, even seedless varieties of things can be made to reproduce. Not using this is a natural vegetative propagation. There is artificial vegetative propagation, where there is cutting, where you're cutting apart and putting it together and making a new plant together altogether. Grafting is there, layering is there. So with the help of that, you can do it. Second thing, it is kind of faster. Than sexual mode of reproduction. So those are the 2 points over here. Offsprings are genetically identical. Offsprings are genetically identical, and therefore useful traits can be preserved. Yeah, that is also one thing. For example, when sexual reproduction is there, male parent gives male gamete, female parent gives female gamete, both of them comes together. Over here, the genetic material that you see are different. But now, if I want a plant like this only, if there is a plant, I see it's pakka good. The flowers are good, the fruits are good. Basically, it is disease resistant also. I want all the varieties of this plant, all the features of this plant to be in the progeny also. I don't want it to undergo sexual mode of reproduction. So what do? I basically cut a part of it which has buds, I keep it in the next pot, now it grows up there. So I have the same type of plant over here. So I am getting all the advantages of the genetically identical of the parent plant, right? So the offsprings are genetically identical and therefore useful traits can be preserved, only useful. So it is rapid and economical method. Then seedless varieties can also be obtained, and seedless plants can also reproduce. That is also one thing. Now name the reproductive and non-reproductive parts of the bread mold. So hyphae are considered to be non-reproductive part of the bread mold. The hyphae has a blob-like structure called sporangia, which is the reproductive part of the rhizopus. So in the case of a rhizopus, it looks somewhat like this. Okay, so over here, this is mycelium, which is formed of so many hyphae. This is also made up of hyphae. These are rhizoids. Okay, this is also made up of hyphae. This is also made up of hyphae, my dear children. This thing that you see over here, this is what? Sporangium. This is the reproductive part, because sporangium holds spores, my dear children. Sporangium basically has spores, and when the sporangium breaks open, the spores are being released, and the spores have the capacity to basically germinate whenever there's a favorable condition. So spores are usually covered in a cyst. So spore is basically very much protected with the help of covering the cyst. Okay, so whenever favorable condition arises, they kind of go sit on the favorable condition. Basically they go land over here, and then for example there's a moist condition, so we have kept the bread out, the stale bread. Now this is a moist atmosphere over there, so the spore which comes and lands over here can give rise to a whole new bread mold like this. So hyphae considered to be the non-reproductive part of the bread mold. The hyphae has a blob-like structure which is sporangia, that is the reproductive part which has the spores. Cool. Always write in points, underline whenever you see important structures like this. Cool. Next, state one drawback of each of the following. Oral contraceptive pills. What is a drawback of oral contraceptive pills? Now oral contraceptive pills, no, it has a mix of hormones like progesterone and estrogen. Okay, it has a mix of hormones like progesterone and estrogen. The problem is when they are in normal level in our body, it's all cool. But when they are not in the normal level of body, it can give rise to side effects. It can lead to hormonal imbalance as well. It can lead to hormonal imbalance. So usually it is being used, basically these are needed for ovulation to happen, that is ovum to being released, and for fertilization, everything for it to happen normally. But in the case of contraceptive pills, their amount is kind of increased. They are being taken from external source also, and now their amount will be totally increased. Now when their amount is increased, they will inhibit all the process they were actually doing. So they will inhibit ovulation. Okay, that is why it is used as a contraceptive pill. But it comes with a drawback, that is it causes hormonal imbalance also in some people. Now copper T is next contraceptive, which is kind of inserted inside the uterus. Inside the uterus, this copper T is being inserted. Okay, there's a T-like structure which is covered by copper, and the copper ions basically what do? They mess up with the motility of the sperms. They mess up with the motility of the sperm. They say sperms you cannot move. If sperms cannot move and go to the ovum, will they fertilize? No. So this is a use of copper T which is being put inside the female body with the help of a doctor or nurse or anyone like that. But what is the problem is that some people could have problems because of the irritations caused because of the copper ions being released over there. So that is again a drawback. Okay. So I told you two drawbacks. Now under which category of contraceptive methods is the use of condom kept? What is condom? Basically condom, for example, you have the external genital organ in male, that is penis. Now it is covered by something. This thing is known as a condom. Now condom is going to act like a physical barrier. Why, children? If this is put inside, this is the female reproductive site, that is vagina. Over here, the sperms, if it is being released, it will stay here in the condom itself. It won't be moving out. So this is going to act like a physical barrier. So the ovum which is there in the female body, within the female body, this and the sperm is not going to meet. So it is going to act like what? So physical barrier. So I'll say this comes under a contraceptive which is known as barrier method of contraception. Barrier method of contraception. Now, in what way its use is better as compared to other methods of contraception? How or why can we say that this condom is better than other methods of contraception? First thing, it is not allowing the sperm and the egg to meet. Second thing, it does not allow the transfer of STIs, sexually transmitted infections, as well. Why do I say so? Sexually transmitted infections are transmitted with the help of body fluids. So over here, since the penis is being covered by the condom, do you think that the body fluids are being exchanged? This is the female body. The female body fluids are not being coming inside the penis. Over here, so the penis, the fluids, it is being released. It does not come in the female genital tract as well. So there is a clear, physical barrier over there which does not allow the spread of STIs also. That is one very good thing about condom. How do you frame the answer? Oral contraceptive pills acts by changing the hormonal balance of the body so that the eggs are not released and the process of fertilization does not take place. As these pills change the hormonal balance, it may cause side effects to the body. That is one effect of them. Copper T. It is placed in the uterus to prevent the pregnancy. It causes side effects due to irritation of the uterus. Now, condom acts as a mechanical barrier that prevents the meeting of sperm and the egg. Condom prevents the direct contact of body fluids during sexual intercourse and hence prevent the transmission of sexually transmitted diseases or sexually transmitted infections known as STIs like HIV, AIDS, hepatitis B, genital herpes and things like that. Okay? Now, the 3-marker corner question, let's get into it. In flowering plants, the pollen grains are transferred to the stigma by pollination, but the female germ cells are present in the ovary. Explain with the help of a label diagram how the male germ cells reach the ovary. So over here, this is the stigma, this is the style, and this is the ovule, my dear children. Inside the ovule, you have 7 cells. 1, 2 and 3. 1, 2 and 3. Okay. 2 nucleuses over here. So this entire thing that I see over here, from here to here, is basically a cell only, and the cell has 2 nucleus. What do you call the cell? I call this cell as central cell. Central cell has 2 polar nuclei. Okay, this thing that you see over here, which I am going to mark it in green, is your female gamete. Beside the gametes, you have the gametes' best friends, which are known as synergids. These 3 cells which are there, they are known as antipodal cells. Now, you do not have to remember everything, just remember the number of cells: 1, 2, 3, 4, 5, 6, and this whole thing is 7 cell. How many nucleus is there? 1, 2, 3, 4, 5, 6, 7, 8 nucleus is there, 7 cells are there. Okay, now listen to me. What happens is that the pollen grains comes and lands over this. The pollen grains will have 2 male gametes, will have 2 male gametes. Later, because of the chemicals being released over here, what happens? The pollen grain germinates over this, forming something that is known as pollen tube. This is what? This is pollen tube. Now the pollen tube comes over here. Okay, one gamete is going to fuse with the female, female gamete. One male gamete is going to fuse with the female gamete, and this is known as fertilization. And the male and the female gamete fuses together to form something that is known as zygote, my dear children. There is one more male gamete. The other male gamete comes and fuses with the 2 polar nuclei that is present over here. So this is a nucleus, this cell also has a nucleus, this also has a nucleus, this also has a nucleus. So basically I will write the 2 polar nuclei plus the second, or basically I will just write the male gamete. the next male gamete, okay? Please understand, it's the next male gamete. Gives rise to, okay, 3 nucleated cell. It will have 3 nucleus now. And what is this known as? This is known as primary, or basically I'll just say the cell which is formed at the end, it is known as endosperm, or the tissue, basically it is endosperm that is going to be formed. So you have zygote being formed, you have endosperm being formed. Okay, what is the role of endosperm? Why it is there? Endosperm is that something that would be providing nourishment to the zygote, my dear children. So over here fertilization happened once, over here second time fertilization has happened, not fertilization, basically fusion of the nucleus and everything has happened over here. So I call this process as double fertilization. What do I call it? Double fertilization. So this is how things work. You have to draw this thing, not entirely, I'll show you what you have to draw. So this is how you have to draw. So show the pollen grain coming over here. This area from where the pollen tube would be entering, you will call this region as micropyle. Okay, so after answering, how do? You draw like this, okay. If you do not want to draw it like this, you can just make a circle like this, you can just make cells. Okay, but for example, the pollen tube is coming over here, that means you have to make the female gamete over here itself. If the pollen tube is coming like this, then you have to make the female gamete over here. Just make sure of that. Now, after the pollen lands on a suitable stigma, the pollen grain absorbs water and nutrients. It produces a tube called pollen tube. Pollen tube grows through the style and reaches the ovary. Its tip contain a tube nucleus and 2 male gametes or sperm cells. The advancing pollen tube enters, okay, so you do not need to write all this thing. Okay, the pollen tube grows through the style and reaches the ovary. The advancing pollen tube enters an ovule, generally through the micropyle, and reaches the interior of the, okay. Advancing pollen tube enters an ovule, that's all, that's enough. Okay, next thing. Here the tube bursts to release its 2 male gametes. 1 male gamete fuses with the egg to form the zygote. The process is known as syngamy, or what do you call this as? Fertilization. The second male gamete fuses with the binucleate, as in 2 nucleated, central cell which forms endosperm. Since the process involves the fusion of 3 haploid nuclei, it is known as triple fusion. So over here, 1 nucleus, 2 nucleus are over here, 1 nucleus is over here. So 3 nuclei is being fused, so you call this one as triple fusion. You call that as syngamy, no? You call this as triple fusion, you call both of them together as double fertilization. Now, thus triple fusion is the fusion of male gametes with 2 polar nuclei inside the embryo sac of an embryo. You do not need to write this also. This, until this is enough. And you have to draw this diagram. Works. So they have gone a bit more to 12th grade level. We do not have to go until there. Cool? Next. List in tabular form 2 differences between binary fission and multiple fission. Okay, what happens when a mature Spirogyra filament attains considerable length? So first we will solve the first question. What do you mean by binary fission, my dear children? In binary fission, for example, this is an amoeba. It will divide itself into 2 daughter amoebae. How many daughters you are getting? 2. In the case of multiple fission, what is happening? For example, this is a cell. The nucleus will keep on dividing. Keep on dividing, keep on dividing, keep on dividing, slowly form a cyst around it. And later, when favorable condition arises, the cyst breaks open and the nucleus, along with a bit of cytoplasm, will be moving out. And now this is what? A whole new cell that is being formed over here. Okay, this is multiple fission where you are getting 1, 2, 3, 4, 5, 6, so many daughter cells there. So binary fission, at the end product is what? 2 daughter cells. But over here in multiple fission, you will get multiple daughter cells. Apart from that, if you see over here, the amoeba, it divides its nucleus and then immediately the nucleus is being surrounded by the cytoplasm, and that's how we get this. But over here, the nuclear division goes on. The nuclear division goes on. After some point of time, the cytoplasm will come and surround it. Okay, after some point of time, the cytoplasm will be formed. Example, you can give of amoeba, you can give of paramecium, you can talk about leishmania. Over here, this is done by plasmodium, the disease causing what disease does this cause? Malaria. It causes malaria. So it is a division of one organism into two similar identical organs. It is division of one organism into multiple organisms. The nucleus first divides mitotically into two, followed by division of the cytoplasm. The nucleus divides repeatedly, producing many nuclei, and many daughter cells are produced later on. So over here the nucleus is divides, and then later on the cytoplasm, followed by division of the cytoplasm. But over here the nucleus keeps on dividing, later on gets surrounded by the cytoplasm. Example amoeba, example plasmodium. Now what happens when a mature Spirogyra filament attains considerable length? For example, this is a Spirogyra filament and it looks like this. Okay, when it attains enough length, what happens is that they will segregate. For example, this broke into this broke into this. Now slowly they are going to regenerate. They are going to regenerate and make newer filaments. This process, first thing, the breakdown is known as fragmentation, and nextly this thing is known as regeneration. So fragmentation is the method, and they are regenerating the lost body parts is known as regeneration. So Spirogyra reproduces asexually by fragmentation. In this, the body breaks up into two or more small pieces or fragments. Upon maturation, these fragments grow into new Spirogyra with the help of regeneration as well. Cool? It's fun, no? The questions are really fun. Now, distinguish between pollination and fertilization. Mention the site and the product of fertilization in a flower. What do you mean by pollination? Pollination means basically transfer of pollen grains. Okay, so basically this is the filament, this is the anther, and over here you have the stigma, style, and ovary. From the anther, from the dehiscence of the anther, the pollen grains will come and land over here. This process is known as pollination. Okay, the transfer of pollen grains from the anther to the stigma is known as pollination. Now, what do you mean by fertilization? Fertilization basically means when the pollen grains land, the male gamete and the female gamete which is there, when it fuses, the fertilization is the fusion of male and the female gamete is known as fertilization. That is the difference. Now, mention the site and the product of fertilization in a flower. So, site of fertilization is where, children? In the ovule, fertilization will be happening. Okay, in the ovule, the fertilization will be happening. What is the product of it? When the male and the female gamete they fuse, what do you get? You get a single-celled thing that is known as zygote. This zygote will continue its division to form embryo. You get it? So, zygote is being formed over here and the place is known as ovule. So, pollination is what? Transfer of pollen grains from the stamen, that is stamen or basically anther, to the stigma. Fertilization is what? Fusion of male and the female gamete. Site of fertilization is ovary. Inside the ovary, what do you have, children? Specifically talking, you have the ovule. Product is what? Zygote. Easy question. Now comes the 5-marker question, and just give me a second. Okay. Suggest any 2 categories of contraceptive methods to control the size of human population, which is essential for prosperity of the country. Also explain about each methods briefly. Can we talk about 2 categories of contraceptive methods? Okay. One is natural method. Okay. Contraceptive methods as in you want to prevent pregnancy, you do not want childbirth to happen. So basically you do not want to conceive, fertilization to happen. So what do? You have natural method, my dear children, where we are not doing it. It's a natural method that is happening in okay, we have some role, but still. Then we have barrier method. Then we have intrauterine devices. Then we have oral pills and everything. Then we have surgical method also. So they asked you to suggest 2 methods and then talk about each of them. Natural method, for example, in that comes something that is known as periodic abstinence. Okay, periodic abstinence. What do you mean by that? Basically, the menstrual cycle is of 28 days. On the 14th day, the ovum is being released. The ovum is being released, and how do baby form? Basically, the ovum and the sperm they fuse. That's how the zygote is formed, and later embryo is formed, and later the baby is formed. So, during day 10 to day 17, if the couple refrain themselves from doing sexual intercourse, okay, during this particular period, then basically naturally the baby would not be found, fertilization is not going to happen. But if you are going to have sexual intercourse during this particular period, then there are higher chances that fertilization could happen. Because I said 14th day the ovum will be released, but it could be here, this time also it could be released, this time also it could be released. In different people there are different time also. You get it? So this is one very important thing. So this comes under natural method. What is it? Periodic abstinence. When during a particular period you are absent, you are not doing sexual intercourse. Okay, I think everything is mentioned over here or not? Not everything is mentioned over here, so I should not be telling in detail, but yes. Okay, then there is barrier method, where we are creating a physical barrier. You're not allowing the sperm and the egg to meet physically. For example, the condom. It is being covering this penis. So basically the sperm is not able to meet with the egg. Similarly, you have walls, cervical walls, which is put inside the cervix. So this is the female reproductive system. Okay, this is the vagina. And over here there is cervix. So there is a cup-like thing which is kept over here, such that the penis, whichever sperm it releases, it does not go inside. Okay, that is cervical vault also. Then there are oral pills, which is given in your book. Oral pills basically is a combination, is pills that contains a combination of hormones, progesterone, estrogen and stuff like that. So in normal level they are going to act normally, but when its level is increased in our body, they prevent ovulation. The ovum release is being prevented. It makes the uterus very unsuitable for implantation. What do you mean by implantation? The embryo comes in and stays there, right? So that process is not happening over there. Okay, now you have surgical method. What do you mean by surgical method, children? Surgical method is when, for example, over here you have the fallopian tube, which is going to collect, the fallopian tube is there, which is going to collect ovum from here, from the ovary. If I cut this fallopian tube and I tie it up, do you think the ovum will be released over here? No. So you're blocking the transportation. But over here the reversibility is very low. You cannot reverse this process. So that is one thing. So what are the things? Methods of contraception: barrier method or mechanical method, condom, diaphragm to prevent the meeting of sperms and ova. Chemical method changes the hormonal balance of the body partner so that eggs are not being released, or basically it makes the uterus unsuitable. Surgical method to block the vas difference in the males, that is known as vasectomy, or the fallopian tubes in the female, that is known as tubectomy. So over here, to prevent what? The transfer of sperm or egg, and hence no fertilization takes place. So this method where I am cutting the fallopian tube, what do I call this as? Tubectomy. Similarly, in the case of males, okay children, in the case of males, what happens? They have a different thing. They have the testes over here. The testes will be having certain areas which are known as testicular lobules, which have seminiferous tubules, which will be making the sperm. So there are guys who take the sperms. There are certain ducts which are going to transport the sperm. For one is rete testis. From rete testis it will go to vasa efferentia. From vasa efferentia it will make a coiled structure which is known as epididymis. From epididymis the things are being taken up to the penis with the help of something that is known as vas deferens. So if we cut over here, it is not getting transported. Things are being made, but it is not getting transported. If it is not getting transported, do you think fertilization will happen? No, it won't be happening. So over here, this process is known as vasectomy. Again, you are preventing the transfer. You are preventing the transfer of sperm and the ovum. Okay. Now, name 2 bacterial and 2 viral infections each that can get sexually transmitted. So there are 2 bacterial infections, syphilis and gonorrhea. Syphilis and gonorrhea, which is sexually transmitted during sexual intercourse, then it will be, there are high risk of it transmitting these things. Viral infection, you have human papillomavirus as well as HIV/AIDS. Okay, you have HIV/AIDS and human papillomavirus at HPV. That is thing. Okay. Now list 2 advantages of using condom during sexual act. I told you, condom basically acts as a physical barrier. Apart from that, it helps in preventing the transmission of sexually transmitted diseases as well. So those 2 things are being sorted. Cool? Works? Chalo, next. Define pollination. Explain the different types of pollination. List 2 agents of pollination. How does suitable pollination lead to fertilization? Define pollination. What is pollination? Transfer of pollen grains from the anther to the stigma. Okay, now explain the different type of pollination. You have two. One is self-pollination and the other one is what? Cross-pollination. One is self-pollination and the other one is cross-pollination. Self-pollination means basically over here you have a flower, you have the stigma style ovary, and similarly you have the anther. From here to here if things are going, okay, in the same flower, you call this as self-pollination. Also, for example, there is a flower over here, same thing, you have the stigma style ovary and you have this also. From the pollen grains from here are going to get transported to here in the same plant. Different flowers but same plant. This is also self-pollination. On the other hand, you have this particular plant also where you have flowers, and from here things are coming to this particular plant. Two different plants are involved. That means you're talking about what? Cross pollination. Works. Now list two agents of pollination. One is wind. Through wind also the pollen grains travel. Other is insect. Through insects also. Insects body, they come to have nectar, and on their body you have pollen grains. The insect go and sit on the next plant and they transfer the pollen grains like that also. So you have insect and you have wind. Okay. And now, how does suitable pollination lead to fertilization? How do you think suitable pollination lead to fertilization? So, where is it? A tube, okay. A tube grows out of the pollen grain and travels through the style to reach the female germ cell in the ovary to cause the fertilization. This is how the suitable pollination lead to fertilization. There is pollen tube that is there, we already studied about it. This pollen grain is there. This pollen grain forms a pollen tube which carries the male gametes, which goes directly to the ovule, and there it fuses with the female gamete and causes the fertilization. So pollination means transfer of pollen from the anther or the stamen to the stigma of the flower. Types of pollination, you have self-pollination and the other one is what? Cross-pollination. So self-pollination means transfer of pollen from the anther stamen to the stigma occurs in the same flower or the same plant. Okay. Now here cross-pollination, pollen is transferred from the anther stamen of one flower to the stigma of another flower of another plant, Works. Done. With this we have done 15 questions and we have included 1 marker, 2 marker, 3 marker and 5 marker questions, which is a great deal, and I hope that you are practicing these questions later at your home also. Now I explain so it becomes fresh, but sometimes I want you to practice it by yourself. It becomes all the more easy to understand things or recollect things. It will boost up your confidence on the day of your exam as well. Okay. And one more very important thing, if you are someone who is confused about what you should be doing after your 10th grade, then here I am for you, we are for you. There is Diksha's Scholastic Aptitude Test, which you can definitely take. What's in there? There will be a 60-minute concept-based lecture, okay? And on the basis of the lecture, there will be an aptitude test. In this aptitude, questions will be asked, and depending on the marks that you are going to get over here, there will be a one-on-one counseling where the counselor would be suggesting the best stream that could be best for you. Okay, and then it becomes all the more easy for you to understand things. So if you're someone who is interested in that, then why think twice? The link is given in the description box. Click on that. And also if you want the PDF, okay, of the notes that is being taught over here, there is a link for that. You can join the WhatsApp channel. You will be getting access to everything. Cool. So that's all, my dear children. Tell me if you have any doubts in the comment section. Okay, the comment section is just for you. I see most of you not utilizing it, feeling really sad about it, but you should be utilizing it. Put down your doubts, we will solve it for you. Put out anything that you want, okay? We would be giving 100% to solve it. And that's all. So wishing you a wonderful day ahead. Bye-bye. Take care.